like, where did it go? Anyway,
That's not good.
I mean, we got what we need for this year. We've already done it. But okay, so get lab on your own this week. Where in the syllabus are we in terms of lab reports? You guys don't know either. Okay, let's look at that. Let's make sure we. Well, you just did one, and we're what we would be is we're a week behind the syllabus because we had to skip a lab, right? So you would want to have, but you're going to have at least 2 weeks of results. I got to look at it. Yeah, let's not worry about it. I'll send you an email if it's something like the Wednesday after break or something. But it may very well be the one after that. We probably get through. Yeah, I'm sorry. Okay, so we have lab on our own this week. I think that's it. Any questions on stuff we've got going on? What?
There's an epic tomorrow.
Epic tomorrow, is that Brezovich? So Dr. B, epic tomorrow in 372. Is it a noon one?
Yeah.
At noon. So I expect that'll be fun. I've seen him give a similar talk before. Those are fun. Okay, anything else? All right. Then we're going to continue talking about fatty acids. What I want to do is actually, let's go back to the notes from the previous day. There's some things we briefly just need to address. Actually, it's a regulation problem. We'll do some regulation problems in a minute. First thing I want to do is I want to talk about, actually this is in the current notes, and this one is not. Let me just move this. And then let's go back to the current notes and let's do a regulation problem there. So when we talk about fatty acid synthesis and beta oxidation, remember they're in different compartments, right? And so they can be regulated in terms of not providing the materials that are needed for a given sort of given reaction. So what we have over here is we have fatty acid synthesis over here. We need to have malonyl-CoA to make it, right? But malonyl-CoA is made from acetyl-CoA, right? Where are you making acetyl-CoA? In glycolysis, where do you make acetyl-CoA? What part makes acetyl-CoA? Pyruvate dehydrogenase complex. And where is that? It's in the mitochondria. And then in terms of beta oxidation, in terms of fatty acid, I'll keep it away. Beta oxidation in the mitochondria, right? Makes acetyl-CoA. So if you decide that you're going to make fatty acids, right? You're going to build up fatty acids for whatever reason, you need to have acetyl-CoA out here, and how you're going to get that out there is you're going to export citrate. Okay. I don't expect you to know this cycle, but if I gave it to you, I could give you questions about regulation. All right. So let's say okay. So this is a regulation problem, and these are reasoning problems. I don't know if we have. I'm trying to think if we have anything that it's like the fructose 2,6-bisphosphate where it's an actual signal molecule we have left. We do have, insulin and glucagon, and what will they do? But in terms of a molecule from now on out, these are all going to be kind of reasoning sorts of things. So how would high levels of malonyl-CoA regulate the carnitine transporter? All right, so what I want you to think about when you see that is What is malonyl-CoA for? You got a whole bunch of it. What is it for? Fatty acid synthesis, right? Remember, we're not going to run a futile cycle. We're not going to run fatty acid synthesis and beta oxidation at the same time. So if we're making fatty acids, right, we're running this, what's going to happen to this? It's not going to transport because you don't want to make fatty acids and then break them down. You make fatty acids and you ship them out of the cell and you store them someplace. Okay? So you see this thing is a cycle, but it's not actually running as a cycle. So it's going to be regulated so that doesn't happen. So if we think about malonyl-CoA, high levels of malonyl-CoA would inhibit the carnitine transporter. You're not going to be transporting, you don't want to transport them into the mitochondria because they're being broken down. You don't want them broken down. You're building them. Okay?
So it's not like
It's not really a Le Chatelier's principle, and for most of these, I'm not going to be asking you to apply some sort of Le Chatelier's principle. It's going to be reasoning, let's take the cell as a whole, and what does it need? But also think about if you look at this and you can say, hey, one of those pathways is on, the other one is off. Okay? You're not running both of these at the same time. Just like you don't run glycolysis and gluconeogenesis at the same time. Does that make sense? Okay, and so we're talking about, so let's try another one. Here's something that's not even on here. How do high levels of AMP regulate the malonyl-CoA, regulate the carnitine transporter? Think about it, come up with an answer, think about your reasoning, and then maybe I'll have somebody share their answer here. Do you have an idea? We don't have AMP on there, but
Like, wouldn't ATP make it go?
AMP?
Or so wouldn't that make? Doesn't that mean you're low on ATP, so it won't
Yes.
break down fatty acids?
Yes. So what's going to happen with the carnitine transporter?
It's gonna happen.
Yes. So how's it? So what it would mean for? So the question is, how do high levels of AMP regulate the carnitine transporter? You got all of it.
It would just make it travel through. I guess
It would turn it on.
Okay.
It would upregulate it. Okay. So if I would take my AMP from over here. Okay. So you have a lot of AMP around means the cell's low on energy, which means the cell needs to break down fatty acids. Let's see. So we can do that for any of these, right? How would, let's do one more. I lost my cursor, there it is. All right, we'll do it here. How would insulin binding to the outside of the cell regulate the citrate transporter? So first off, to remind everybody, what does insulin signal?
Activation of cycles.
So what is it? What is it signal more generally? Yes, it's going to activate glycolysis, but what more generally does insulin being in your blood mean? Low blood sugar means high blood sugar. Okay, insulin's released after a meal, means you have high blood sugar. Glucagon is the low sugar one. Okay, so we're getting signal that we have high blood sugar. But let's assume we're going to assume this is a liver cell because liver cells do everything. Uh, muscle cell is not going to be doing fatty acids. It's not its job. I mean, I guess they have fat in that tissue though, but I don't think I don't think muscles are doing any of that. In fact, I'm almost certain. So insulin binds the outside of the cell. Just had a big meal. What's going to happen here?
Make pyruvate.
What was that?
Make pyruvate. Run glycolysis.
You're going to run glycolysis, but what's going to happen to the citrate transporter? It's going to activate it. You've got lots of energy stuff, so you're going to store it.
Okay.
Right?
In your body.
Does that make sense, the way you think about these things? Think about the energy state. If you have lots of energy, you store stuff, right? You make glycogen, you make fatty acids. Okay. In terms of glycolysis, you have insulin, right? So you got insulin, so you're gonna run glycolysis, you're also gonna make glycogen. You run glycolysis, you make pyruvate, it gets broken down to acetyl-CoA. If you've got enough ATP in the cell, that acetyl-CoA can then be made into citrate, shipped out, and help make fatty acids. In terms of talking about that, do you see what our path is from, you see our path from sugar to fatty acids to fats? So, before I painted my door, I had a fun comment that, I have one of those metabolisms that takes all the, takes the excess sugar I eat and turns into fats for storage, or something like that. He said it a little bit better. It was like one of those, it was a pun on I have one of those metabolisms where I can eat anything, right?
So it used to be citrate and carnitine, like to pass through because like acetyl-CoA just like can't pass through by itself.
Correct.
Okay.
Correct. Okay. And when we think about the mitochondrial membrane, right? I don't know exactly where these are, but they're probably in the inner membrane. The outer membrane on the mitochondria is fairly porous, like fairly big things. Like I think it's 5,000, something about 5,000 grams per mole can make it actually through the outer membrane. But the inner membrane holds out protons. So that's a super tight membrane. Okay, so that's kind of just an example of regulation sorts of problems, and they are reasoning problems. They're almost always reasoning problems, except when we have the very specific things like that. All right, so this is just kind of what we were talking about. This is some more written stuff on that. Okay, so you can take a look at that with the notes. So we've so far talked about, in terms of fatty acids, we've had it's all been saturated fatty acids, right? So what happens when we don't have saturated fatty acids? Well, if the double bond is in the right place, that it just comes in. We've talked about this before, that it could just fit into beta oxidation, right? We're making the first step of beta oxidation is we're making a double bond. And so as we're chewing off these 2 carbon units, we encounter that double bond in the right position that it just fits in, then it just keeps going, which isn't what's going on here. It would have to be like that, okay? So if the double bond is in the wrong place, there is a mechanism to handle that. So if we have unsaturated fatty acids, that's going to be moved by this enzyme, this isomerase. Okay. You just need to know this. I'm just mostly pointing this out that you know it exists. Okay, so there are mechanisms, but what would this be an example of? I have this thing, right? I've got a bunch of fatty acids. Some of them are unsaturated with a double bond in the wrong place. And So things happen and then they can go into beta oxidation. What's that an example of? In catabolism.
Convergence.
Convergence, right? Catabolism is convergent. You try and get all the molecules into the same pathway as much as possible, right? And make as many of the same, and the whole beta oxidation process is making acetyl-CoA, right? So the thousands of different fatty acids that can exist, may not be thousands, but they may be. Now, they all can eventually be funneled into beta oxidation and made into acetyl-CoA. All right. Also, though, if you're doing we've talked about even fatty acids, right? So even fatty acids, what you can do is you can chop off 2 carbons at a time and make it all into acetyl-CoA, and then there's nothing left. But what if there's an odd number of fatty acid carbons, right? Because that happens, not a lot. Well, this simple pathway right here takes care of the last 3 carbons. Okay. So I'll blow it up, but you're not going to need to know that either. But I do want you to be aware that, we've got these other things to take care of odd cases, we might call it. If this pathway exists, though, it's not like a xenobiotic thing. You know what I mean when I say xenobiotic?
No.
It means it's not something that's natural to your body. So it's something, so when we think about your liver, right? Your liver takes care of drug metabolism, so you take drugs and they have to think about what your liver is going to do with this, or they can think about it, and they'll have some ideas. They also have to do some experiments to make sure that that's true. It's not super easy to predict, and it's also very different in each person, right? So that's what they worry about. They'll sometimes worry about some drugs work on some people and not on other people. Okay, sometimes it has to do with how your liver's metabolizing them. So xenobiotics, your liver is set up to take in whatever weird things you have, and I'm going to say detoxify them. Mostly it's about making them water soluble so you can get rid of them in your urine. Okay. But this is not xenobiotic, right? Your liver developed to do that when you grew weird berries out in the woods when you were our caveman ancestors. It didn't do it because you're taking, Xarelto. But so here we've got 3 carbons, right? So what would happen here is beta oxidation is going to come in, and it's going to get us down to those 3 carbons. This is propanol-CoA. So beta oxidation. Okay, so we'll just say when. I'm not going to write it. Let me type it. Okay, so when odd-numbered fatty acids need to be metabolized. Okay, beta oxidation. I'm going to say breaks them down until 3 carbons are left. Okay, those 3 carbons, are going to be then metabolized by a separate pathway. Let's say catabolized. All right, and I expect you to know that. All right, and we'll just very briefly tour the pathway. So we've got a propanol-CoA here. All right, we have some more of these fun reactions where to get the reaction to happen, to break something down, we add a carbon dioxide to it first. Okay, so we got a 4-carbon thing. We're rearranging it. We end up with succinate. Well, then what are we going to do? Right, this happening in the mitochondria, right? We're at succinate. All right, now we have a pathway. We can now take it to fumarate, right? That's just using the citric acid cycle. You can then take it to malate. And I don't think do you guys remember malate and pyruvate? Okay, so we now have a pyruvate, and now we're right in the region of what we know how to do things, right? So pyruvate, we can, I guess in here it's going to make it an oxaloacetate. No, it's making it acetyl-CoA. So pyruvate, we can just take through the pyruvate dehydrogenase complex and then acetyl-CoA and run through the citric acid cycle. So I don't expect you to know it, but you need to know there's a separate pathway. But see, this should look fairly familiar, right? We're going to get it into succinate, then we're in the citric acid cycle. We can run around the citric acid cycle until we get malate, and then we can pull it out and make pyruvate. Did we talk about that enzyme briefly? That it's a way of making pyruvate. You'll make malate and pull it in. You'll make oxaloacetate, or you make pyruvate oxaloacetate and pull it in.
We talked about that it was possible, but I don't think we would have gotten that.
Yeah, I think I didn't emphasize it that much this year, how we get things in. And you don't need to worry about it. Yes, the actual sentence. You see that? That's what you need to know. Okay. But hopefully looking at the rest of it, you're like, oh yeah, okay. Your cell is using existing pathways as much as possible for all these things. How can its natural selection has made it such that how can it shove this thing into here or here and not have to make new enzymes? Ignore that. Goodness. Yeah. But I'm hoping it would warm my heart if you're recognizing all these places that we've already learned that things go.
And does that happen like all in the biochemistry?
All of this bit?
Yeah.
Yes.
It doesn't have a travel mesh from the when it's like
Right.
recarbon to the citric acid.
Right. So the beta oxidation to get it to this 3-carbon intermediate would happen to the mitochondria, right? Okay, it could ship it out and do all this stuff before the succinyl-CoA, right? I succinate, but succinyl-CoA is in the mitochondria, or is in the citric acid cycle.
Yeah, that's what I want.
That's in the mitochondria too. So it could ship it out and bring it back in, but I bet pretty heavily it doesn't.
Yeah.
Right. That wouldn't be very efficient. But so we get here. There's that GTP, right? That we get to see in the citric acid cycle. There's our ubiquinol, right? But we go. So it's all in the mitochondria, and all the way down here. Everything here is inside the mitochondria. This is the pyruvate dehydrogenase complex. Okay. It looks like no. This is that. Okay. Well, I did have it there. Let's go on, and we're going to start talking about cholesterol and cholesterol metabolism. Okay. So this is cholesterol. It is an important part of membranes. Okay, it affects the fluidity. We talked about this last term. So it makes membranes more fluid or less fluid depending on the context, not complex, depending on the context and what's needed for those membranes. So if you're a very stiff membrane, let's say really long fatty acid chains, I think we didn't talk about lipid rafts last time. Lipid rafts will be made up of lots of. We did talk about. Oh gosh, forgetting the name. We talked about sphingosine. So you think about your sphingolipids, they often go in these lipid wraps and they're really stiff. And if you needed to loosen those lipid wraps up a little bit, you could stick some cholesterol in there in between the molecules. If you instead had a membrane that had a lot of double bond fatty acids in it, that would be pretty fluid because they don't stack very well. And you could put some cholesterol in there to help shore it up. So it really depends on the context. It's also a precursor for the steroid hormones, which we talked about last semester, and we also did talk about our bile salts, okay, that aid in digestion of fats. That's what this is needed for, the main uses of cholesterol. And so you have to get cholesterol around your body. You talk about worrying about your cholesterol, right? And when they're worried about cholesterol, they're worried about certain complexes that we'll talk about, or macromolecules, molecular assemblies, let's say. You need cholesterol, okay? Too much of it is bad, but you do need it. And you need the good and the bad types of things. So what we're going to talk about here then is, we talk about, you've heard of LDL and HDL probably, right? In terms of types of cholesterol. And what these are, they're these lipid complexes that are delivering things around the body. Okay. So in terms of thinking about one of these complexes, we have proteins in them. The proteins end up being part of the membranes. They help signal, right? They help interact with various cells and let them know what kind of complex they're looking at and what kind of lipid protein they're looking at. These are called lipid proteins. But they're carriers. What we have is we have phospholipids around the outside here. The tail's pointing in. It's not a bilayer. And then inside, we're going to have a bunch of triacylglycerols and cholesterol esters. All right, so I want to remind you, so cholesterol, why is this necessary? Well, how soluble does this look in water? Your blood's mostly water. Not very, right? When it goes in the membrane, this is its polar head, which I always think is kind of a silly polar head. Not very big deal, right? It's not charged. One hydrogen bond for the whole molecule. But everything besides this is nonpolar. So these aren't very polar. If you want to get them around your body and get them dissolved in blood, or, well, carried in blood at any reasonable concentration, they get packaged like this. So that we have like a liposome, but it's, so we package them in this lipoprotein, is what this whole complex is called. Triacylglycerols. Let's look at those. So, right, when we have fatty acids have, a charged end. So they're not super soluble, but if you remember our triacylglycerols, how polar does that look? More. Okay, but you've got some hydrogen bond acceptors here, but really not very polar, right? You put them, you add these to the glycerol, you've got this really non-polar thing. And this is, when you think about we've got this complex over here, but also when you think about your fat cells, your liposomes, not liposomes, adipocytes. So when you think about your adipocytes, right? They are storing fats like this, not like fatty acids, because it's a nice big fat glob. It's really nonpolar, actually. All right, we can just do hydrogen bond acceptors. Esters are, it could be worse. It could be like an ether, but they're not polar at all. So that's how you're going to carry your triacylglycerols and your cholesterol esters through the body in these things. All right, there are 4 of these that we are going to talk about. Actually, we'll go ahead and take a look at this picture for that, and then we can talk about each of these individually. All right, so I'm going to do these in kind of chronological order about how the lipids would run around your body. So when you think about ingesting fats, what happens? They're broken down in your intestines, and your intestinal cells are going to make what's called chylomicrons. Okay, and what these are going to package up cholesterol and fat to go to the liver. I'm going to say triacylglycerols for fat. And what I'm going to say is actually, let me say instead of package, they're not packaging them. They are the package. Made by intestinal cells, made in the intestine. All right, so when we think about these chylomicrons, they're right there. That's this. We'll go with that. We'll color code it. All right. Questions so far? So you're making this big old complex to kind of transport stuff through the blood. Yes.
So you said triglycerols, they're good, they're better than, they're good at transporting what? You said something earlier.
I said there. I was talking about how polar they are and how soluble in water,
Yeah.
and it's better. Esters are more polar than ethers, but they're still not all that polar on their own.
Okay.
Is really what I was saying.
So you're saying you need these things to transport the non-polar things.
Right,
Okay.
and in fact, that's the method, right? So it's not that you accidentally get triacylglycerols. What actually happens is, let me do this. You have triacylglycerols. We'll do tag. Okay. You've got this. If you think about your intestinal lining here, you've got tags here. They get broken down into fatty acids to take them across the membrane, and then they get rebuilt up into triacylglycerols to send them to the liver. So it is a very intentional thing. You don't take triacylglycerols across the intestinal lining as triacylglycerols. Bridget?
I don't even know what I'm thinking, so.
Okay. All right. If it comes to, let me know later.
Sounds good.
But really, what I want you to know here is this. So chylomicrons are going to take these things to the liver. The liver is going to absorb the chylomicrons, and it's going to make a new one, which is called a very low density lipoprotein. This is LDL. It's going to become LDL, but not yet.
So it's going to, whoever's going to take it in. What are all the other random letters that are just there? There's LPL and CATPA, and then P and LCAT again.
They might be enzymes. I actually don't even know what they are. I used to have this really cool Flash video that did this, but then they outlawed Flash. Yeah, it's Flash.
Yeah.
Yeah. So, and I wasn't smart, and I didn't record it in some way. I could have screen recorded it, but I lost it.
So when they
So in other words, what I'm concerned with here is let's just not worry about. Let me find a big black pen. Okay. Not worry about that or that or that or that. Bile is fine. Bile salts go to the intestine and help out, right? I'm not going to erase it. Since we've talked about that, I'm not going to worry about that. And we're not actually going to either going to worry about these bits. You don't need to know about that. I'm solely concerned with knowing what chylomicrons are, what they do. I want to know how all these are made and where they go and what they do. Okay, but chylomicrons, very low density lipoprotein, and high density lipoprotein. Okay. Yes.
How does this relate to what we were just talking about?
We are talking about lipid metabolism.
Mm-hmm.
And so we have switched from talking, we've scaled up to kind of, when people talk about where does biochemistry end, we're scaled up, we're right up at the top edge here, right? Because sometimes we're talking about a cell, what's going on with a cell, but we're getting all the way up to organelle size things. But this is about lipid delivery in the body.
So we've moved on in our like big diagram from That, like the
That
fatty
this
acid.
this would not be in there at all.
No, okay.
Yeah, that's it. So the big overall diagram for metabolism, this is about shipping things around in your blood. So good questions. Thank you for asking me to clarify. All right. So the liver will then make. Let me take some to the liver. Let me add where they are absorbed. Okay. The liver's going to repackage those triacylglycerols and the triacylglycerols and the cholesterol as very low density lipoproteins.
Wouldn't it be like high density though?
No, so the density protein is more dense than fat, than lipids. And so right now we're going to have a lot of triacylglycerols and we're going to have a lot of cholesterol and not so much protein. So the liver will repackage the triacylglycerols and cholesterol as very low density lipoproteins. Here, let me go ahead and abbreviate them up here. So VLDL, which you don't really hear about. I'm trying to think, I just had a blood test. I should have looked and see if VLDL showed up. LDL shows up, HDL shows up. All right, so the VLDL is going to go around in the blood and it's going to deliver mostly triacylglycerols to tissues. It's possible there's some cholesterol transfer there, but most of the cholesterol transfer is going to happen with LDL. Okay. All right, so once enough triacylglycerols get pulled out of VLDL, it's now considered LDL. It's more dense because we pulled triacylglycerols out and a higher fraction of it is protein. Okay. So people talk this transition. This happens just by pulling out triacylglycerols. Let's go ahead and write that. Let's actually make this. Where'd my highlighters go? I can highlight in orange. Let's go with the orange here instead. So that's this. Okay.
You said it's not classified as LDL.
Oh, it is classified as LDL. Sorry. All right, so that's just a transition of stuff coming out of it. All right, the LDL binds to specific receptors. It binds to the LDL receptor in cells. And so that cells that need cholesterol are going to internalize that. They're going to absorb it. Okay, and then it's going to be broken down for use in the cell. All right, and so that's here. Yes.
And it's still the chylomicrons that are shipping after delivery.
It's not the chylomicrons. So the package is from here to here, the package is chylomicrons. Okay. From here to here, the package is very, very low density lipoprotein.
Is that what cholesterol is considered?
So when we think about what they say, bad cholesterol and good cholesterol, this is considered bad cholesterol, having too much of this, and this is considered good cholesterol.
Because it's too much fat for the LDL. Is that why it's fat?
I'll talk about what happens with this. If you have too many of these things that are around your blood too long, they stick to the blood vessels, and then they cause an immune response, and that's how you get your hardening of the arteries and stuff. I think I'll talk about it today. We only have about 14 minutes left, but okay. So, Daniel, did I get your question answered? Okay. So there's separate delivery vehicles.
LDL is just its own protein that ships the
LDL is its own lipoprotein. It's one of these. Okay. And this one with the actual things, I don't remember which one this is, or even if it was labeled. This is an example of a lipoprotein. Part of what will define it is these particular proteins in it. But it doesn't matter in terms of from our perspective. All of these are kind of this. Chylomicrons have more fats and triglycerides and cholesterol, as do LDL, as do VLDL. LDL, you've got less of the triacylglycerols in here, and it's more dense in the cholesterol, and then it gets absorbed, and then it's gone. Okay. So one thing that people often get confused on, LDL is not going to become HDL. Okay. We haven't talked about HDL yet. All right. So and VLDL or LDL is low density lipoprotein. You probably got that. But okay. High density lipoprotein. So when you have your test, your high density lipoprotein is the one that they call the good cholesterol, and the reason for that, so that's going to be here, and then I'll go ahead and get rid of those two other arrows for our purposes at least. So high density lipoprotein, it's made in the liver, and its role is to go through the body and sweep and collect excess cholesterol from cells.
If it's not made from LDL, why is the arrow from?
It's not. It goes through the cell. Oh, the two in the middle there.
Is that what you're saying?
I said I was going to get rid of those for our purposes.
Okay, I heard you say it. I just didn't see it.
Oh, it's a highlighter. It needs to not be a highlighter. It's made in the liver, yes. So here, it doesn't exist.
Perfect.
Clearly from the figure it does exist, but we're not going to worry about it. Okay, so its role in the body is to go through the blood and collect excess cholesterol from the cells and bring it back to the liver. No, cholesterol, the molecule. That's why it's confusing, because they call it good cholesterol and bad cholesterol, but it's not cholesterol. This is cholesterol. This is these are lipoproteins. There's cholesterol in the lipoproteins, exactly. Okay. But medically, when you go into the doctor and things, they're talking about the lipoproteins like they are cholesterol. Okay, they contain cholesterol. So its role in the body is to collect excess cholesterol from the tissues and return it to the liver. And if you got too much cholesterol, your liver is going to break that down. Okay. We're not going to talk about that pathway. Questions on what those four things are doing? Yes?
If you have a lot of cholesterol, does your liver make a lot of the HDLs?
It is not my impression necessarily that that's the case. I don't know what happens with the metabolic disease, but you exercise, you get more HDL, right? But it seems like if your liver was just going to respond and make more HDL when you had too much, people's levels seem to move differently. That's just my kind of real life, what I've heard sort of thing. We'd say that doesn't happen. So if you exercise more, you end up with more HDL. This has been correlated with better outcomes. Okay. This has been correlated with worse outcomes. Okay, but what's interesting is this is where you always want to look at the meta-analyses. Is when this isn't actually a meta-analysis. There are meta-analyses out there that say, hey, yeah, these are correlated with better. If you take drugs that lower your LDL, and you can take those, okay, so you can control how fast these are made. But if you take drugs that lower this, that's correlated with better outcomes. So when I say better outcomes, I mean fewer heart attacks and lower mortality. A decade ago, they came out with some drugs that actually raised this, and then they did the meta-analyses, and it didn't matter. So this is correlated with fewer deaths, etc. Right? But this is clearly not directly causing it. It's something that's we'll call it a side effect. And what may have been going on to some extent is they talk about, hey, yeah, we filtered out for amount people exercise. We filtered out for health-seeking behaviors. If you exercise a lot, you're going to have high density of lipoproteins, and you're going to be healthier, right? Because of the exercise, not necessarily because of the HDL. And so when they do a lot of these studies, and they're not meta-analyses of randomized, let's say they're not randomized control trials. They say, hey, we filtered that out. Well, they're not as good at filtering that out as they might lead you to believe. Okay, I'm not saying the scientists are lying. They know what they're doing in their statistics and stuff. But then when people analyze those, it's not as good as it was. And did I talk to you guys last time about or last semester about the estrogen studies? So it used to be estrogen you could take as a woman, you could take estrogen post-menopause to avoid heart disease, right? So heart attacks and things like that. And so they had like 400,000 women worth of observational studies, right? People are essentially reporting what they do, and they tried to filter it all out and look like they had this really strong effect. And then they went and they got some randomized control trials down, and it turns out it's a little bit worse. If you're taking estrogen, you've got a slightly higher risk. So there are you want to see the randomized control trials.
Wasn't it because like the people who could take estrogen were like wealthier, so they could like.
You can hypothesize why that happened, right? You're gonna have, if somebody can afford to take estrogen, they're going to be able to exercise more. They're not probably going to be working 2 jobs. I mean, there are a whole bunch of things. They're probably getting on average better sleep, right? Lots of stuff. Okay. The main point here is what though is get those 4 Lipoproteins, you need to know what they do. All right, so then let's go ahead and here actually is an example of I could talk about some disease states here where we have deficiencies in terms of cholesterol metabolism, and the two main diseases are related to either taking up LDL. Okay, so you can imagine if you've got our LDL here, and it's going to come in, it's going to bind. You may have talked about this in biology, platinum-coated pits. Okay. But you have, they will bind the outside of the cell, you'll end up with a pit, it'll come into an endosome, and then it's going to be broken down into its parts. So we're going to break down to get amino acids out of it, cholesterol, any triglycerols that might be left will be pulled out as well. All right, and so one of our diseases, which is familial, if I don't have it written down below, I'm not going to remember it. But it's a defect in these receptors, the LDL receptors, where you don't pick these up. And what you have, and it's a genetic disorder. And what happens when someone has that, I'm not going to, it's in the study guides, but I'm not going to make you responsible for it. But the example here is when you will have 5-year-olds that are having heart attacks based on the fact that they can't take up their LDL, or they can't take up their LDL well. They have high concentrations of LDL, and they end up with heart disease really, really early. Okay. I don't know if I have a picture for Tangier's disease. Okay, we have that next. So here's if you have one of these, if you have that sort of deficiency, or if you have a lot of extra LDL, you can get these fatty spots. So especially the pictures I see for them are really, I see them in the eyes for that, but you get this deposition of lipids because you've got so much LDL going around and it's not being taken up. And I don't know how dangerous that is. The other disease, though, let's go back to this picture. It's not on this picture. It's called Tangier's disease, and it's a defect in the ability to flip. There's a flipase. Do you remember what flipase is? Okay, flipases will take cholesterol. There's a flipase that'll take cholesterol from in here and put it on the outside of the membrane, and that's how. And then HDL can totally pick it up as it runs by. And Tangier's disease, that flipase doesn't work, and so cholesterol can't get out of the cells. And it's another example of where you have things like 5-year-olds with heart attacks. Okay, so very important that you get these LDLs taken up by cells, and that when cells have excess cholesterol, that HDL can take them up. Let's see, 2 minutes. I think I could talk about how this is related to heart disease really quick. I think that's the next figure. We'll skip that. Here, we'll do this. Okay, this is a simplified figure. You can see there's a whole lot of complex stuff that happens here in terms of making plaques, and especially because there's an immune response involved. But essentially, you've got your artery here, and if you have a lot of LDL, it starts to deposit on the side of the blood vessel. What that does is that, and what's not shown here is you're going to recruit an immune response as a result of it. It ends up being irritated. You end up with inflammation, and you get a bunch of white blood cells that are going to pile up in here then. And then they essentially pull in calcium, and you end up with calcified. So you hear about hardening of the arteries. You get calcium in here in terms of the plaque. Okay, and then where you have a heart attack is here they're showing you're getting a blood clot in here. So you get something, if you get a blood clot that forms here, your blood clot swims around and it finds its way to one of these places that's restricted, and then it stops. Okay, you can have that happen. If that happens to the heart, that's a heart attack. It'll cause a heart attack. And the problem there is that your heart either tissue gets deprived of oxygen. If that happens in the brain, then that's an I want to say aneurysm, but aneurysm is a expansion, right, of a spot.
Yeah.
It's a stroke. That's when you get your stroke is caused by blood clot in the brain.
Oh, yeah.